Hello, my name is Travis Monk. This is one of a series of videos involving bacteria and viruses. In this video, some of the major classifications of eubacteria will be described. While animals may have obvious physical differences between them, that's not exactly the case with bacteria. For this reason, many of the ways that bacteria are classified may seem a little bit peculiar. The only physical characteristic that bacteria are commonly classified by involves their shape. A bacterium that belongs to the genus Staphylococcus is shown on this slide. An explanation for that name of this genus will be provided later in the video. While there are countless different bacterial shapes, three are pretty common: cocci, bacilli, and spirilla. Cocci are spherically shaped bacteria, such as the example shown on the left portion of this picture. The singular form of the term is coccus. Bacilli are rod-shaped bacteria, as shown in the pink bacteria in the middle of this picture. The singular form of this term is bacillus. The last common basic bacterial shape are spiral or spirilla. These bacteria are often described as corkscrew-shaped. Sometimes bacteria live on their own, independent from others. Other times, the bacteria form long chains or large clusters. The image on the left shows a cluster of round-shaped bacteria. Because it is found in a cluster, you could use the prefix staphylo to describe bacteria organized in this manner. The particular group of bacteria would be referred to then as staphylococci, since they are made up of round-shaped bacteria. Staph infections refer to any clustered groups of bacteria that cause disease. They're pretty common. The image on the right shows long chains of round-shaped bacteria. Due to the fact that they're stuck end to end in a chain, you should use the prefix strepto to describe how they're organized. This group of bacteria could be described as streptococci. Strep throat is the result of bacteria such as these shown on the right. Again, bacteria are pretty small and simple. For this reason, trying to classify them can be a challenge. As a result, the following few forms of classification are not based upon physical structures, but more creative means. Earlier in the year, we discussed photosynthesis and cell respiration, as well as the organisms that do these different cell processes. Autotrophs were described as producers, or organisms that make their own food. The term auto means self. And heterotroph are used to describe organisms that eat others. Hetero means other, for survival. These two terms should look pretty familiar. The only new bit of information involves the different ways that organisms can make their own food. When we discussed photosynthesis, we were describing a process that photoautotrophs, or organisms that get their energy from the sun, hence the photo, do. A very brief overview of this process is shown on the left. Chemoautotrophs use chemicals, hence the prefix chemo, to make their own food. An example of this process in a deep sea vent using sulfides is exhibited on the right side of this graphic. Bacteria can also be classified by the means in which they use oxygen. Some bacteria, just like humans, need oxygen to survive. These bacteria are called obligate aerobes. When you see the term aerobe, think aerobic exercise or aerobic respiration, where oxygen is needed. Obligate looks like obligated, which means required. Some organisms find oxygen toxic. It kills them, just like humans find carbon monoxide toxic. These organisms are referred to as obligate anaerobes. The only difference between this term and the last is the an. As a reminder, a or an in front of a word means not or without. They are required not to have oxygen. Finally, some organisms fall in the middle. They don't need oxygen to survive, but they do just fine with it. They are referred to as facultative anaerobes or facultative aerobes. The last way that bacteria can be classified may seem a bit obscure. You're just classifying them based upon the color they turn when you add different color stains. But it's probably the single most important, and it's referred to as gram staining. The reason that this means of classification is so important is that it often can be used to determine how different infections can be treated. All eubacteria possess a peptidoglycan layer that's made of sugar and protein in their cell wall. In these pictures, the peptidoglycan looks like a bunch of sausage links. In some bacteria, gram-positive, the peptidoglycan is a thick layer that's found on the outside. This is shown on the set of top illustrations. When stained using a pretty simple procedure, gram-positive bacteria shown up purple, or blue under the microscope. In other bacteria, gram-negative bacteria, there is an outer membrane that surrounds a much thinner layer of peptidoglycan. This is shown in the illustration on the bottom. When gram-stained, gram-negative bacteria show up pink or red in color. Gram-positive and gram-negative bacteria also react differently to antibiotics, disinfectants, produce different toxins. So again, this knowledge has important medical applications. You may treat a gram-positive bacterium with penicillin, but it might not help a gram-negative infection at all. That is the end of this video involving different classifications of eubacteria. If you're interested in learning more about bacteria and viruses or any other topic involving biology, please subscribe to my channel. Thank you.